If I could invite the speakers to join us at the table up front. And if folks want to use the microphones at the two end to questions you might have. So let me start with one. Lynn, I was going to begin with your talk on the pancreatic cancer. Given all the work that is done in this space by other organizations and agencies and whatnot, NCI, et cetera, why do you think that an organization like yours needs to actually do what you do for your particular disease, as opposed to thinking about it in the broader scale? What is the advantage?
Although there's a lot of similarities across cancers, there's also a lot of distinctions between them. And so in the case of pancreatic cancer, there's either specific backbones that we know have some efficacy, and a lot of the activities that are ongoing in the field are single agent activities directed towards a very targeted therapy and a specific indication. So in our case, we believe that we have to marry what we know about pancreatic cancer, what we know is efficacious in that disease, with anything new that we want to test in the field in terms of biomarker-driven therapies. And so we think that it's that putting those together for this specific patient population, it leverages what we know in others, and yet it takes advantages of the distinctions that we know about this specific population.
Okay. So let me, David, if I could, if you think back at how AMP came together, can you sort of remember what might have been sort of key deal breakers that essentially had to be overcome to get all of these parties to engage? In terms of giving us a little bit of open the curtains for us in terms of the process of how the sausage was made.
So I can think of two early decisions. So actually, the initial discussion about data access was a little bit contentious. The NIH wanted to make everything available as rapidly as possible. The companies said, if we don't have at least a little period of exclusivity here, then you get a kind of free rider effect where other companies are coming in, they're not investing what we're investing. We would like some period to look at these things. And so that's where we got the 6 months from. And as I said, it's kind of turned out, maybe because we have the example of ADNI in the Alzheimer's space, for example, everything's out there within 3. So it's really not been much of an issue. And I guess the other was the funding mechanism. And this was an interesting conversation. Again, the question was raised, well, should we allow biotechs to be part of this? And, they don't have any money to do this kind of thing and invest at this scale. And again, the companies pushed back a little bit and said, they don't have any money today until, they go for their IPO, and then I have to buy them. And they cost an awful lot. And, we don't want to have a situation where, again, we're funding, what essentially is something a VC or, a business development activity here. So the rule has been that if new folks are going to join new initiatives, and we do have a couple that we're thinking about in different disease areas, or people want to join an existing initiative, they need to come in with something that's a comparable scale.
Nadeem?
Thanks. I thought I'd be consistent in asking an awkward question first up to each of the panels. Chaz, that was a great presentation. And you reminded me, when I first joined the industry from academia, I was introduced to 2 phrases I'd never heard before. The first was the mysterious deliverable, and the second was the tangible measure of success. I'm still not entirely sure what they mean, but they're 2 things I'm asked to think about frequently. And the 5-year deliverable and measure of success for the Bromo Domain project was very striking. 14 clinical trials, a company valued at $500 million. And he said you had more than 30 such endeavors. So in that light, I wanted to ask David and Meg, what are the measures of success of your proposed and ongoing initiatives, 3 and 5 years? And then ask Lynn to react. is that what patients want?
I thought we were going to have to arm wrestle, so who went first, but you're very kind. Um, to me, the measure of success is that I have pages and pages of questions that would make a difference for the targets that we could start to pursue. That I think that this type of data would that the EMR linked biobank data would allow us to answer that we cannot answer now, and you know, we don't have enough money, we don't have enough we don't have time, there isn't the collaboration need, you know, we don't have enough resources, the N is a problem. So to me, one of the measures of success would be being able to quickly scope and perform studies in a way that would have an impact on drug, you know, discovery in the timelines that we're expected to develop drugs. That would make a huge difference.
So ours are very specific to each of the research initiatives within AMP. So in the type 2 diabetes case, there are clear phases to this, as I showed you, and there are clear milestones along the way. The go, no go milestone is essentially that we have a functioning portal, that there are specific things that it can do. So there's a specific list of functionality, that we have X number of data sets with X, you know, from X type of sources that are in that, and that it be able to answer some, you know, questions. When we had our first review with the extended EC, they said, you know, we asked what should the measure of success be, and one of the heads of R&D said targets, you know, that we can use, that work. And so they're very specific. You know, in the case of the Alzheimer's disease initiative, half of this is this putting tau imaging into clinical trials. Those are 5-year trials. So we won't have the readout on those till the end of 5 years. So that's a whole different kettle of fish.
So in terms of the patient perspective, I think patients would appreciate those as measures of progress, but not necessarily measures of success. So from a patient's perspective, you know, success is quality of life and you know, longer life, those kinds of very meaningful changes in health or disease or, you know, whatever the issue is. And so I think it's recognized that these are all steps of progress towards that goal, but what they're really looking for is something that's going to get over that ultimate goal of what really in their mind, what they're looking for is for real success, not just progress.
Yeah.
I
Chaz,
was
you wanted to respond?
anticipating you would say that, and I was going to ask, given that anticipation, I was going to ask Chaz to react to that. So how can we move from, I really like the way you phrased it, measures of progress to measures of impact?
So let me share with you what we've just agreed with Myeloma UK. So what they've agreed to do is to take our 37 novel inhibitors, epigenetic inhibitors. They're going to test them in myeloma cells from patients. If anything looks exciting, they're then going to work with a CRO to take our molecules and convert them into clinical candidates. And then they're going to fund a clinical study in an academic setting. So in nowhere in that process have I even mentioned big pharma. So I'm not saying that we shouldn't work with pharma, but I'm just saying that this is a new model. This is being driven by the patient organization. And what we've set ourselves a goal is that within the next 5 years, we're going to come up with 3 new targets for myeloma. And we'll create a pipeline around them.
So Chaz, I actually have a question, not a follow-up per se, but a slightly different one. So every time I hear you speak about what you've done, I just wonder if there's any world problem, we should just hand it to you and you can solve it. But presumably this wasn't easy. And it's a sort of similar vein question of, you know, what I asked David. You know, can you share a little bit about how you got all these parties to work together? What do you need to do to ensure that these kinds of partnerships can be successful? You know, you're acting in a sense as, I think the word that was used earlier in the day, perhaps as an honest broker, and perhaps that gives you a certain currency to be able to transact. but perhaps a little bit more detail on people who are thinking about perhaps using your model or building upon it, that they can understand what the drivers for success were in being able to achieve as much as you have in 5 years.
So I'm often asked that question, and I always give the same answer. And I would put it down to three things. Firstly, the Wellcome Trust in the UK have, over the past decade, Mark Walport, now Jeremy Farrar, they've really driven this open science, pre-competitive data sharing, etc. They have really championed that, and they have been sort of big charitable funders of ours. The second thing I would say is, and Lon's standing there, but I think GSK has actually been a leader in the industry. Tashi Yamada more than a decade ago, more recently Patrick Valens, now Lon, these guys realized, and we heard it from Meg, this is just so damn difficult. We can't do it on our own. No one organization, no one institution, no one group can do this on their own. We're going to have to pool our resources to try and increase the probability of success. So credit to GSK. The third thing I would say is in Oxford, 800-year-old university, Lon, you've been there, we have the same challenges around incentives, publication, promotion, et cetera, But we have a chap named John Bell, who's our Regius Professor of Medicine. And I don't know if any of you have ever met him, but the guy is an absolute visionary. And he has really allowed us to do this. So I would say those three things have really enabled us to do this.
So I'll just focus your question, because I was going down exactly that path, and it was going to be directed at you, Chaz, in the first instance. When I want to be difficult within the organization that I work in, I ask the pharma side, what do they bring to the party? Right? With all this specialization that's going on in academia, what are we bringing to the party from pharma? And people get a list of responses to that, but at the top of that list, almost always, is molecules and chemistry. So with that, and that's as true today as it was many years ago, I think. From that lens, how did you get past a situation of getting people to share their molecules and expertise when that is crown jewels of the pharma? And what can we learn here from the SGC's lessons, as well as others that I think others have been involved with in this audience that have been, the scars must be there. And can we avoid them this time?
Well, all I can say is that my sense is across the industry, more and more organizations are realizing that they can't do it on their own. So our Epigenetic Pro project, it was started with the Wellcome Trust and GSK, but since then 9 other companies have joined. And many companies, they keep refunding us because they say the one thing they recognize about our outputs is that they are reproducible. Our colleagues at Novartis have repeatedly said this. Everything you guys do, we trust. And, be it our structure, any data with our molecule, et cetera, et cetera. And of course, there's a big issue at the moment around sort of, and there's been some high profile publications about the reproducibility of some literature, et cetera. So I don't know whether, Lon, that answers your question. I'm sort of, I think, I absolutely believe the pharmaceutical industry is brilliant at certain things. It's brilliant at things that require scale and infrastructure. It's brilliant at high throughput screening, lead optimization, toxicology, regulatory, doing the really big clinical studies. What none of us is any good at is target discovery. But I also believe that there are certain things that are much easier to do in an academic setting. I'm not saying academics are better. I'm saying it's easier to do in an academic setting. And that is working with clinicians, working with patient groups, accessing patient material, and accessing public databases. That's important.
It's Arthur Holden again. And just to add on to Lon's question, and Chaz, your commentary was, and David's was, builds on this model of bringing people together, going after large challenges in a way that no party can really do independently. Or they may be doing that, but they're not going to do it to the scale. And we've heard this commentary as we've gone through about pre-competitive, I don't know what the other side of pre-competitive, competitive, but in putting together these types of activities, and particularly when you have many large companies and small companies involved, the guidance that I've received over the years in this area is to ask a fundamental question. Is the activity that you're engaging in pro-competitive? not pre-competitive. In other words, is what your output, and just take Chaz or even David or what Lynn is doing, what that is helping is that anyone and everyone that's interested in working in this area can be more productive by using this platform that's being developed. Legally, that's the only thing that matters from an antitrust point of view. If it passes that test, and just by way of context, is a number of years ago, I think it was in the 70s, the Congress in the United States passed the National Cooperative Research Act, which actually was put in place to protect the information technology industry so that they could work together in industry-based collaborations to create these pre-competitive platforms that would help competition down the road. And so there's plenty of precedent for doing this type of work, but I'd like to just add one comment in terms of the success factor, because I think if you look at FNIH and having the NIH involved, or the activities that Chaz has done and I've done over the years, I view having an entity like the Wellcome Trust, with its orientation towards open science and improving the productivity and results orientation, if everybody's being really critical, I would agree. But I would also highlight a subtle point, and maybe the panel can comment on it, and I'll step back. Is I think in putting these types of initiatives together, it's really, they're risky. They are risky. And so putting together the right plan that has reasonable objectives, of which you meet, ideally exceed, and build confidence in the leadership and the collaborative structure that's put together, really makes a huge difference in terms of building on the success. So it's not of any surprise that Chaz is sitting there and has been funded many times. Results beget more results, because, so it's, I'm just be curious in terms of your commentary. You said nothing about planning, but obviously planning is really important. Have the right plan with the right contingencies and then make it happen.
So one thing we always do is at the start of any funding session is we agree some sort of quantifiable output. So it could be so many structures, or it could be so many inhibitors, or it could be so many TEPs. It could be now we're testing molecules in these new assays. So it's going to be new human assays. And again, these assays we're going to share with the world, etc. So they've always been quantifiable, and the funders like that. But of course, what we always try and do is exceed those outputs.
Meg, actually, could I follow up a question for you, since you're at the sort of the early stages of this? As you listen to all of this and the early conversations you've had with your partners, can you share, to the extent possible, what are the hurdles you foresee that you would need to overcome to make your plans a success? What do you need to tackle?
I mean, this is a great opportunity as we really begin, what I'm sure is going to be a tough road. I think actually the conversation that we had this morning around getting the results data sharing model right, so that the EMR linked biobanks are protecting the patients, so that we're able, that the groups that are coming together are able to ask the questions that have so much value, and that people are able to meet their own objectives, publications for academics, the type of outputs that are being important for industry. So I really think, in thinking about it, the biggest challenge is protecting the patients, at the same time allowing us to really utilize the data in a way that is going to make a difference to the type of questions we'd like to be able to ask.
Other questions from the audience? That side of the room is completely quiet. Encourage you to get up to that microphone, but why don't you go ahead?
Yeah, Jeff Ginsburg. I'm going to change gears a little bit. So obviously data has been a common theme for everything we've talked about today. The data sets are massive and complex, and I wanted to ask you about how we're going to analyze this data to come to fruition. After all, there's only one Russ Altman, and we have lots of data to go around. Now, seriously, I particularly, Meg, when I saw your game-changing, view of the world, I'm wondering how you're thinking about bringing to bear the kinds of skill sets that are going to be required to really analyze the data, and then how do we create a force amplifier of that, to go across all of these programs that we talked about today? Because to me, it seems like we're heading towards an incredible bottleneck to really create value from the data. And I can tell you from the academic perspective, we're still very challenged in how do we generate the next generation of analytic minded data scientists to really address these types of challenges. So I'm asking you what you're doing in the industry.
I guess I think there's a couple of aspects. I mean, I think there's definitely a data harmonization aspect that we really need to consider. I mean, one of the driving principles is that no one resource is enough, and every single company that's part of this consortium has gone through the pain of giving really exciting results and then not being able to replicate them, not being able to really generalize them in a way that convinces you that it's real and allows you to really move beyond that. And so I certainly think that'll be an important aspect. I think the concept that you walk before you run, I think we want to start with some of the if you look at that arrow of what we can do now, we want to do a really great job at associating genetic variants with outcomes. Then we want to look at progression. And so I think we're imagining sort of starting a little and moving that on. But I think the most important thing, and I think part of the reason, and we had a really good debate about this at lunch, that we say that no one group can do it by herself, and this kind of concept of duplicate, doing a lot of different companies, kind of doing the same analysis at different times. And there certainly is value in, multiple groups looking at the same data set. But I think what we're thinking is that if there are some ways that we can get the economies of scale and look at outcomes in a very comprehensive basis, the quicker we can move to focusing on longitudinal data analysis, what would that really take? And I feel like, I think that the consortium that we're attempting to put together will not be a success if we just do at a larger scale what we can do now. The key thing that all of us have said, in those conversations, is we want to be able to look at information over time. And so I think there's really a sheer, how can we kind of quickly move over, quickly take the economies of scale for what we can do now and move to the next step.
David, you'd wanted to comment earlier?
Yeah, so I think to answer the question you just asked, there's a couple of different things. The first is having quality partners to build the data sets and to do the analysis. So I probably should have mentioned, of course, of ours, the portal's being built at the Broad, and the ability that they have, the deep understanding of the genetics and the IT aspects of this, and their deep networks of relationships with the type 2 diabetes community has really been essential to success. And having, the second thing is that you want to do pragmatic, practical analyses to the point that was raised earlier. What are you going to do for patients? Well, the fact that you've got industry involved, they're not just bringing their molecules, they're bringing their viewpoint. Probably what patients don't need right now in type 2 diabetes is another glucose lowering medication. And so we're going to be focusing, when we do these targeted genotype phenotype analyses, on complications, on things that relate to beta cell function, and something that's going to bring forth a new class of therapeutics that really is going to be novel and not something that's just a me too on what we already have. And then the final thing is that we make everything broadly available. So really, we're not doing all the analysis. It's all out there, and the entire biomedical research community, including the companies, are free to go forth and develop anything they want out of this, including novel IP.
One follow-up question there, David. In setting that up and releasing the data, did you define common data elements that you need to follow in terms of how the data were generated and submitted by the various parties?
Yeah, so there is a data model across all of the type 2 diabetes, and it really depends on the analysis. The network analysis of the human brain samples that's being done in Alzheimer's, really, we're not doing sort of those kinds of integrated queries, and so there we're using Sage Bionetworks. We're using more of a collaborative workspace platform that allows folks to share their analyses and do much more of a discovery-based type of activity. So yes, in some cases we definitely have them, in other cases not so much.
Yeah, hi. Meg, I'm gonna ask you a question, but I'm hoping everyone can comment on this. So the resource that we're hoping to build How do we ensure that it lasts, that it's not just something that we do for the duration of our tenures at our companies, that there's something there for everyone else to take advantage of when we're done? And I think the SGC has some examples, and there are other examples, but how do we make something like this last?
Yeah, I don't know the answer. I mean, I think, I think on the one hand, what really drives a resource like this is your need to be able to use it, and its relevance to the questions that you have, and so I think that the key thing is to continue to challenge yourself in terms of I think that the biggest danger is you build something and then basically the technology moves beyond, what's there, and then it becomes something that's not useful anymore. So I certainly think that's one aspect is to evolve it in a way that continues to make it a critical aspect for the research that you do. But I also, please comment, because you guys have actually, looked at the problem of how do you continue to make sure that it is useful and that it lasts.
And I'm not sure I know the answer to that, but one thing we do is before we release any of our reagents, we actually put them through a peer review. So we have a scientific advisory board, which comprises external academics. So we will have already shared it with our pharmaceutical network. So we'll have got their critique and so on and so forth. But before we release it to the world, we put it through an external scientific advisory board, and their job is basically to assess the quality. So it's not just me saying, oh, we've got an inhibitor. They're saying, look, this inhibitor, it's potent, it's selective, it's cell active, and you will be able to use this to dissect biology. I should also say, in relation to that inhibitor, what we're now doing is generating, for the same target, we're generating multiple chemotypes. So they're active molecules, but different structures. So of course, if you get the same answer, then of course you can be confident that the effect you're seeing is on target. The other thing we do is we're also now generating what we call negative controls, so inactive compounds as well. So you have a positive and a negative. So hopefully with that sort of toolkit for each target, you really can work out, does this target do this in this assay?
I think, the sustainability is going to be related to its success. I mean, if it succeeds, then you will find a way to evolve, a way to continue to keep it funded, keep all the partners playing, and go forward. So not that that's necessarily easy, but I think it's really, the basic answer is just make it work and you'll be able to continue it.
I think one of the great failings of public-private partnerships is that so few of them have exit strategies. Now, in some cases, that's okay. They're meant to be open-ended, and as you say, the partners will decide, we've had enough. In the case of AMP, we said from the beginning that sustainability was going to be one of the issues that we would have to be deciding on, but I think we fairly decided that until we saw what we had built and knew what we had, wasn't wise to tackle that. So we're at the halfway point, and each one of the co-chairs for each of the steering committees has been tasked to come up with a sustainability plan, which includes when are you going to sunset this resource? How long is it going to be used? If you're going to transfer it, where are you going to transfer it? And what's the additional incremental cost to do that? And then we will have a discussion at the extended EC about who's going to pay for it.
Maybe if I could just add to that, I think sort of, these consortia take a long time to build up. But once you've got a network that is working effectively and trusts each other, then ideally it's good to try and keep them together somehow. So with the SGC story, initially the organization was started to generate novel human protein structures, do them as quickly and as cheaply as possible, and share them with the world. A few years later, we started working, that's largely soluble proteins. A few years later, we started working on membrane proteins, which of course were a lot more difficult. A few years later, we started using these structures to design small molecule inhibitors. Now we're using them to sort of build human assays. We're generating these target-enabling packages. So all the time we're evolving, but to be honest, there's so many things we need to do. I think once we've got a network of friends, then let's just carry on doing it. I never worry about funding. I believe if we're doing the right science and if we're having an impact, funding will take care of itself.
Tim?
So I was just going to make an observation to the question on sustainability. To me, as sustainability is what you've just described, Chaz, that constant evolution. And I think the constant evolution comes from recognizing that there isn't a right and wrong way to do this. I mean, clearly you have 37 different leads. You're not being informed by human genetics to have generated 37 leads in the timescale. So you've gone back to, as a medicinal chemist, we can generate ways to perturb the system in a very controlled way, which will inform biology. It's an experiment we need to continue to do. All of these are different types of experiments that are trying to get to the same aim. The one thing I would say about this meeting is I don't think we should try to get to a solution that sort of says this is the best thing to do. I think we should be encouraging an environment that enables us to do all these experiments.
Yeah, just a quick add-on in terms of, I think one of the advantages of this type of collaborative model is that it's okay for it to go for a period of time and then stop. Think of most commercial entities. That isn't okay. That's failure, when in fact in this model it could very well, it depends on the question you're answering, what you're going after. But as an example, 8 years and looking at all the major serious adverse events and doing the genetics on those, we're probably getting to the point where we've accomplished what we need to accomplish. And the byproduct is the data and the scientific insight that Lon was highlighting in his chart with it. But I do think it also gets back to this risk and diversification approach. Even if you take a project like Meg is summarizing, which by the way hasn't been planned and laid out yet, it needs to have a plan and figure out exactly what it's going to do with it. But the idea that you might find after a series of well-conceived pilots that it isn't the right thing to go forward, or based on that data and information, you may say, here's the right way to evolve it. And that's what I think where Chaz, you've been doing your work on the targets. You can see you've done a certain amount of work and then you realize, yeah, this can happen, even the way you pick the targets, how you evolve to that. So it's very initiative dependent.
And just add to that, I mean, there's some talk this morning about sort of publications and promotions in an academic setting. So I sit in a very competitive academic university, and we produce about one publication every week. And I should also say that probably all of our publications, they involve many of our industrial partners. And Malcolm Skingel-Lon at GSK has done this analysis that publications which involve industrial partners usually have many more citations. So that's worth bearing in mind. Secondly, promotion was mentioned. I've been back in Oxford 8 years. In the past 8 years, 8 of my PIs have been given chairs in Oxford or permanent positions in Oxford. Maybe that just says it all. So I think the model can work in an academic setting.
So just in the last 10 minutes of this session, I wanted to steer us towards an area that we haven't quite discussed yet in the field of alternative business models and entrepreneurship. And that is this idea of energized entrepreneurs being drivers of innovation. And I wanted to ask Lynn to react to this first and invite the others as well. So thinking about the power patients have in driving entrepreneurial innovation. An example, we recently met with a group of patients who had advanced stage cancer, and we were discussing with them, what is your unmet patient need? And to a person, they all said, it's not really what my need is. If I could ask you to do one thing, it would be to make a medicine that can prevent cancer. How many pharma companies currently are working on drugs to prevent cancer? Probably not that many. And part of the reason for that is, as part of that process, we then did an internal analysis of, let's say, we had a idea that inhibiting enzyme X could indeed prevent breast cancer. How big a phase 3 trial and how long a phase 3 trial would be needed to demonstrate that we can prevent breast cancer? And it was very big and very long, prohibitively so. So we won't even entertain that concept. I wonder, and that's just a kind of an extreme example, but I wonder what role patient groups could play in directly expressing these are needs that we have that are currently not even being explored, let alone met. And part of the reason for that is the regulatory framework isn't right, the process of doing the clinical trials isn't right, the way you're doing the science isn't right, and then use that to stimulate entrepreneurs to react to that. And if patients can drive that kind of innovation, the others can comment on, once we know that, what can we do about it to meet it?
Well, I think it's amazing how when there are challenges put out broadly across the world, through social media or whatever, whatever platforms there are, there's an amazing amount of response to those challenges. And so it's a matter of figuring out, I think, how to harness that there is an interest. It's defining a problem and finding people who like to solve problems, who kind of, jump on and try to figure this out. So it's a way of getting a collective interest, incentive that is not, not necessarily in the market, although that might be downstream, but something to get it going initially in terms of what those ideas are, and then convert it from that, I guess, from that social to that market, kind of activity. So it's probably a progression along those lines. Get excitement about it, and then turn it into the opportunity.
Maybe ask David for your reaction. Given, how can we leverage the millennials and the kind of entrepreneurial spirit that has kind of reemerged in the kind of post-Facebook generation, and apply that towards drug discovery?
So I think that intrinsically all of these efforts are doing something like that, because we're either all making, patient resources or data or tools, reagents and so forth, publicly available at no charge. So, I mean, automatically what you're really doing is lowering the barriers of entry for, individuals, for companies, and for entrepreneurs to take advantage of that. So I think that's inherent in what we do. There are other interesting things you can do with large amounts of data. There are these incentivized challenges and so forth, which I think further democratize the discovery process. But I think really just kind of creating, you've got to have a lot of data to do those things. That's what attracts people to those kinds of challenges. So creating those large data sets, making them broadly available, creating the resources, making them available, I think really is a pretty key first step.
I think the other thing, and I think maybe you mentioned at the beginning, but I wanted to reiterate, I think clearly articulating the question. I love, with the first slide that you put up is saying that we want to reduce the cancer risk by half by 2012, by 2020. And how, I can imagine how that drives all of your funding decisions. I can imagine how that vision is so clear. And I think that was one thing that was really crystal clear as the consortium group put together. I think that we had so much frustration that really trying to articulate the things that were driving us was so critical. And if we could really kind of have that very patient-centric vision that articulates, I think that is very helpful in addition to the aspects that you mentioned as well.
I suppose all I would say, Nadeem, is that sort of, what patients want is they want novel medicines, they want effective medicines, they want affordable medicines, and they want them quickly. Even tomorrow is too late. They want them today. All of us, I think, we get our inspiration from patients and carers and relatives. But, and we've all got our own little stories. But, I've had, the dean of our medical school got esophageal cancer last year. He was operated on in July. 2 days before the operation, he rings me up and says, Chaz, I want you to take my cancer cells and test your compounds on them. This is how desperate patients are. I've had a mother of a daughter with a rare gastrointestinal stromal tumor, a GIST. And so this is 1 in a million. It's intractable to existing drugs. She sits in my office and she's crying. And I say to her, sort of, how's your daughter doing? And she says, my daughter's 22 years old and she's just had the whole of her stomach removed. I'm sitting there, how the hell do I respond to that? I gave a public lecture in Oxford last September, and this mother comes up to me afterwards and says, Professor Bounter, my daughter died of glioma earlier this year. If you want to study her brain, this is where you can get it from. And she hands me this scrap of paper with her daughter's name, her date of birth, the day she died, and where her brain was stored. So, all I would say is that patients, their relatives, their carers, they are desperate. So I think if there's anything we can do, we need to inject more urgency into our discussions.
So given that we're close to the time, I sort of reminded of, won't bring any Donald Trumpisms into this, but thinking about a previous political arena, you can maybe end with saying, it's the patient's stupid. So we'll stop there.